Hello everyone, my name is Eduarda Nassif Gomes. My project is Antagonistic Action of BACILLUS VELEZENSIS against Phytopathogenic Fungi of Corn. My advisor is Ligia from the Genetic Department of UFPR. So, contextualizing my project, corn is a very important vegetable in humans and animals' diet, so everything related to this culture is important to society. There are some diseases that cause the rot in corn that are specifically caused by some phytopathogenic fungi. Normally, farmers treat this problem with a commercial fungicide, but as science has developed, they have observed that there are some bacteria that has the ability to inhibit the growth of this fungus. In my study, I have observed the antagonistic ability of Bacillus velezensis that is classified as a plant-promoting growth bacteria that, besides having the plant-promoting growth, also has the antagonistic ability, as studies have shown. I have tested this against the four fungus, Fusarium verticilioides, Coledotricum sp, Fusarium graminearium, and Stenocarpella sp that caused the rot in corn in the field. So, the aim of this study was to analyze whether or not Bacillus velezensis has the antagonistic ability in vitro to inhibit the growth of the four fungus, Fusarium graminearium, Fusarium verticilioides, Stenocarpella sp, and Coledotricum sp. This was averiguated by testing microbiologically and molecular this bacteria and fungus. So, my materials and methods. First, the microbiological test. I have done the pear culture, in which we use a agar done with potato and dextrose. They would display on the petri dish in one side the fungus and the other side the bacteria, and after that observe, after some days, observe the growth between of them in which the bacteria inhibits or not the fungus. So we have done 5 treatments: the negative control, in which is just the fungus to observe it grows by itself; the positive control, that we observe the growth of the fungus against a commercial fungicide, compare this after that with the bacterial growth. The fungicide we used was Vitavax, and the other 3 treatments were about the bacteria, so LGMB12, LGMB319, LGMB 426. That are 3 isolates from the microorganism collection from the Microorganism Genetics Laboratory of UFPR. The other tests of microbiology that we have done is the nitrogen fixation test, in which we used an NFB medium that has a green color, and we would place 5 dots of the bacteria. If it has growth and has the ability to fixate nitrogen, it would grow in a clear circle. If not, it would stay the same. The molecular tests, we have done PCR, but first DNA extraction, that we confirm the quality by the equipment nanodrop and electrophoresis. And after that, we have done the PCR and electrophoresis to confirm. We have tested that for the genes of the NRPS domain, surfactin plus iterine, and surfactin P, that are genes that codificate antagonism in bacteria, especially in Bacillus velezensis. The results for the paired culture were really positive. We could observe that bacteria has inhibited the fungus better than the fungicide, and that it showed in the graphic, and also in the qualitative results that we can observe in the plates. On the other side here of the graphic, we could observe some examples of that. The results for PCR were positive only for the NRPS domain and surfactin gene, and the other one, they were not positive, maybe because the bacteria don't have the gene. For the nitrogen fixation test, all the results were negative. Concluding, from the statistical and qualitative results, can be said and confirmed that the Bacillus velezensis can act antagonically against the phytopathogenic fungi, and can be a promising and viable option of biocontrol in farms. So the next steps of this research would be to evaluate that em vivo. These are the references and thank you.